This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1059, recorded on November 3rd, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Hello, everybody. And let's see. I'm looking at the window. I've actually stuck my head out. It's a little coolish, but it's a beautiful autumnal day. It's in the low 60s. The humidity is low. The clouds are puffy and friendly looking, and rain on the way.
Here it's 14C and sunny. It's very sunny here in the city.
Yeah. Oh,
Also
and Sunday is marathon day, by the way. But just wanted to drop that in there too.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 56 Fahrenheit, 12 degrees Celsius, moderate breeze from the southwest, and it's totally overcast.
So joining us from Austin, Texas, Rich Condit.
Hi there. 75 degrees, partly cloudy. It's gorgeous.
It's gorgeous.
From Western Massachusetts, Alan Dove.
Good to be here. 55 Fahrenheit, 13C. Pretty much like the clouds Dixon described. And other than the leaf blowers going on all day, it's lovely here.
And from Montreal, Canada, Angela Mingarelli.
Hey, everyone. It is maybe the coldest here, as per usual. It's 11C, which is like 52 Fahrenheit. It's overcast, just like where most of you are at. Is it sunny anywhere except for where Rich is?
It's kind of it's pretty sunny here.
Okay. And we actually had snow 2 days ago. There were flurries in the morning. It's melted already, but it's begun.
We had a little bit a couple of days ago, too.
Okay.
We did too.
If you like our work here on TWiV and all the other podcasts of Microbe TV, please consider supporting us. We need your support to do this. Go to microbe.tv/contribute. American Society for Virology. Hey, maybe Kathy, you should read that. Sorry.
Sure, that's fine. ASV is again providing the Global Scholar Travel Awards, and this is to international virologists. If you're interested in attending ASV 2024, which is June 24th to 28th in Columbus, Ohio, you can look into doing this. Preference will be given to junior faculty presenting abstracts. The reason I'm talking about this now is this year the application deadline has been moved earlier to November 30th, 2023. It's less than a month away. This is to ensure enough time for the recipients to obtain visas. You don't have to submit an abstract now for this Global Scholar deadline, but if you indicate that you're going to do one, that might give you a little edge, and the abstract deadline then will be the same as for everyone else, which is February 1st. And the link for this is asv.org/ASV2024/global-award. It's not a very convenient link, but I haven't checked it out. But I think if you just go to the ASV website and click on the meeting, you may be able to search around or find the Global Award application. You don't have to be a member to apply, so we encourage that. And I just got back from another site visit in Columbus. It's going to be a really great meeting. And Dixon, there's a really cool thing in the Greater Columbus Convention Center, which is the first ever vertical farm inside a convention center.
Yes!
So
Nice.
I took a picture, and I'll send it to you later.
Oh, thank you so much.
And
That's
this,
good news.
this is the Global Travel Award, so it's for people outside the U.S. There's also a regular travel award for ASV, right?
Correct. It's only the global one that has this early deadline so
Right.
that they can get international visas in time. Yes, thanks.
Thanks to all of you who participated in our Halloween fundraiser at vaccinated.us. We'll let you know how we did. I have to get
Rich,
the
your
data from
picture was amazing.
Matt.
Everyone's
Yeah, Rich.
picture was, but Rich was like especially amazing.
So if you have pictures of you in your capes and spike shirts, please send them in. We'd love to see them.
And remind us where we can look at those.
Well, you can't.
Oh,
Actually,
I thought there was
They're on
discussion about where you
Instagram.
could see them.
They're on
They're
Instagram.
Okay.
Microbe TV. How is the Instagram? Let's see. Instagram.com. Is it Microbe TV without a period? Let's check.
They're also
Microbe.
on X, well, Twitter, X. I saw Rich there.
Yeah, they're on
Social
God,
media.
I was on
Instagram.com.
X.
The user is microbe.tv. I see. Even Jolene sent one in. We have Mark Martin. There's Rich. We had Daniel, and we had Cindy, and myself. So lots of activity. So if you have one you want to share with us, send it in. We'll blast it out on all the social media, or if you don't want to, that's fine too. Just let us know. But thank you for your support as always. And of course, this is November, December, January. For 3 months, Parasites Without Borders is raising funds for Microbe TV. You can go to parasiteswithoutborders.com, make a donation. Daniel will match it and give it to Microbe TV for the months of November, December, and January. It's a good chance to give a nice chunk of change, and it'll get matched.
How about that?
Okay. How about that? And don't forget, in Amy Rosenfeld's laboratory at the FDA, the Center for Biologics Evaluation and Review, there is a research assistant position available to work on enteroviruses, pathogenesis, cross-reactive antibody responses, developing animal models, all BSL-2 work. You can go to the show notes. We have a link where you can read more and have an email to inquire more information about that. And speaking of
Yeah.
Amy
In fact, if you're
Rosenfeld.
curious about the cool types of work you could do in Amy's lab,
Yeah, that's our snippet
listening.
for today. It's at Nature Communications. A human monoclonal antibody binds within the poliovirus receptor binding site to neutralize all 3 serotypes. So the poliovirus receptor binding site is dear to me. It's on my phone. It's on my cell phone. Okay, because I have a structure of poliovirus bound to its receptor. You can see exactly where the receptor is binding around each 5-fold axis of symmetry. So this is very cool. It's an antibody that binds in that site. But the authors here are, let's see, do we have co-first authors? Does anyone know?
I don't think so.
Okay, Andrew Charnesky is the first author. Then Susan Haffenstein is the last author. And in there is Amy Rosenfeld and Kostya Chumakov, who has been on TWiV a couple of times. And this is from Penn State, of course, the FDA, and a few other places as well. So I think this is cool because a monoclonal antibody had previously been identified. It's called 9H2. And it was identified from, I don't know if I'm supposed to say this or not. So Kostya Chumakov has gotten so many polio vaccines in his lifetime. His mother gave him OPV every 3 months in the Soviet Union so that we'd be partially protected against other infections. He got like 60 doses of OPV in his lifetime. Okay.
That's crazy.
So he donated his blood to it, and they isolated from his blood, lymphocytes, and they screened for antibodies that would neutralize all 3 serotypes. So that was the screen, and they found this one.
But that's not included in the paper. It's
a separate publication.
Extra detail. Extra detail.
we should have done this experiment in your lab, Vincent.
Yeah, with my blood.
Because we were always getting, inocula of stuff. Confessing to sloppy lab technique here, but, everybody's vaccinated. You're not worried about it. You're handling the virus. Yeah.
Also, the fact that it was accepted on the 17th, received on the 19th, within 2 months they already published. This is within the same year. That's extremely fast. Like within 2 months they already got published.
It's received
2022.
2022. I'm blind.
So a year later it was
I'm
accepted.
blind, sorry.
No, it's just, that's more typical, right?
Yeah.
Fix
That
my
was
contacts.
another brownout.
Yeah, literally, yeah.
So just to remind you, there are 3 serotypes of poliovirus, 1, 2, 3. There are wild types that used to circulate. Now just serotype wild type 1 circulates. There are 3 Sabin vaccine strains, 1, 2, 3. There are 3 inactivated polioviruses, 1, 2, 3, and they all bind the poliovirus receptor, which we called PVR, and then was named by others CD155. I kind of like PVR because my initials are in it, right? Poliovirus receptor. So that was cloned by Kathy Mendelson in our laboratory in collaboration with Eckhard Wimmer many years ago. And the receptor binds in a groove around the fivefold axis. And because these viruses are built with icosahedral symmetry, there are 5 copies of each subunit around each fivefold axis. And there are 5 binding sites for the receptor. So a total of 60 binding sites for particles. So if you look at my phone image, this is an image where we took poliovirus and mixed it with soluble receptor, and there are 60 binding sites in the reconstruction. So
And so that sort of structure that's at the fivefold axis, there's a peak in the middle, and that groove around it is often referred to as the canyon. Is that term still correct? I think they use that term.
They
used
used
in
it
the
in the
paper.
paper.
yeah,
Yeah.
they use it. Now look, this is a model I had made.
Oh,
yes.
wow.
This is a 3D printed model of poliovirus. So there's the receptor in blue, and you can see, I think, around each fivefold axis, there are 5 copies very nicely.
And they're
This, by
sort
the
of diving into the canyon, right?
Yes.
They're dipping into the canyon there. Yeah, exactly.
Like Luke
right.
Skywalker. Yep.
And what's cool about this model is it comes apart.
Woohoo!
So it can uncoat.
So it can
Look
encode.
at that. Look at
And
that.
I could put genome in.
You've got Ann Palmenberg's
Or
genome around
M&Ms.
somewhere,
You
right?
should put M&Ms in there instead.
Make it a pinata.
I have Ann Palmenberg's
Exactly.
genome, which is hanging behind me there. She beat
It's-
it for me.
Yeah.
It's not actually Ann Palmenberg's genome. It is
No.
a genome of poliovirus that Ann
That Ann
Palmenberg gave you.
Palmenberg gave
All
us, yes.
7, however
Just to
many,
be clear.
Yeah.
7,000 nucleotides.
Well, the cool thing is that, let's see. So there are 4 different bases, 4 different colors. And at the 3' end here, you can see a stretch of like 30 red ones. So obviously A is red. Right? So she did a great job. So this antibody, they wanted to know how this antibody could neutralize all 3 serotypes. The capsid, previous work with monoclonal antibodies, identified 4 different sites that monoclonals are directed against on the surface that neutralize infectivity. And so they said, well, where is this monoclonal binding? So obviously the thing to do is to solve the structure. And these days you do cryo-EM because you can do it very quickly. You don't have to make crystals. And they solve the structure of this antibody together with wild-type poliovirus and Sabin polioviruses. And you can see the antibody sits right over the canyon, right over the receptor binding site. And, there's a lot of nice images here showing that. But this is a perfect snippet because that's the story.
Yep.
There's actually one more experiment here.
Figure 1 is basically the whole point of the paper.
Yeah, you got
It's great.
you got wild type 1, wild type 2, Sabin 3, and you can see the receptor in blue is sitting right around the fivefold axis where the
Actually, I'm
the
going
monoclonal antibody.
I'm going to say figure, what is it here?
Oh,
6?
right.
Figure 6, I think, C, where it shows the footprint.
Yeah.
And it shows the footprint. I really like this. It shows the conserved antibody binding contact sites in the footprint. It shows in blue, shows the non-conserved ones in red, and then it shows the contact sites for the receptor outlined in cyan.
Yeah.
And man, they overlap, but it's not completely overlapping.
Right.
And the monoclonal site is not completely conserved. So this is a real stunt.
I have 2 comments about this figure, or somewhere, maybe it was one of the earlier ones, where they call it a quilt diagram. And I really like that image. And now I forgot
I've never
the second
actually
point.
seen this before. This is the first time I've ever seen this footprint.
It's basically the surface laid flat with each amino acid outlined, and then you can color in the ones that are contacting either the receptor or the monoclonal.
Yeah, it's a really cool way
Michael
to
Rossman
visualize.
made this popular many years ago.
We are visual animals, and this absolutely plays into that. If you don't understand anything about the chemistry, you still understand the way this thing works. It's really amazing.
Now,
It's
another
great
key
visual.
experiment here, besides the cryo-EM, is the competition, which really shows that if you take soluble poliovirus receptor and you ask, will that compete with the monoclonal antibody. So
Sure.
they take either the monoclonal or soluble PVR. You add it to virus, which is stuck on plastic. It's anchored on plastic. And if you put monoclonal on first, then you go in with PVR, or if you put PVR on first, you go in with the monoclonal next. So the monoclonal can outcompete soluble PBR that's bound to the virus already. So if
Huh.
you bind soluble PVR, the monoclonal can bump it off. Higher affinity.
Wow.
I remembered the second thing, which is this is open access. So you
Yes.
can go look at this. And I found where they call it the quilt. It's in figure 5. The roadmap of the virus surface is represented as a quilt of amino acids.
Mm.
It's nice. It's very nice. And so the other thing here that's important is, so that this is the binding with soluble PVR shows basically that the antibody and PVR are competing for the same binding site. That's the mechanism of neutralization. Another possible mechanism would be that the antibody changes the conformation of the particle. Maybe it has it so that the RNA pops out, right? So when you add soluble receptor to poliovirus, the virion opens up and the RNA comes out and it loses infectivity. So the monoclonal could have theoretically done the same thing, but they do experiments to show that's not the case. There's no conformational change. Rather, it's simply steric inhibition.
It just gets in the way,
Gets in the way.
and it can actually displace.
Yes. Yeah, the antibody can displace PVR, and PVR cannot displace the antibody.
It's just a nice, elegant, like so many papers these days. I start reading it, I'm like, oh wow, digging into this and that and the other thing, and I'm finally trying to synthesize. In fact, the second paper we get to today is a lot, and there's a lot of data here, and they did a lot of work, but you get to the point you're like, oh yeah, of course, this is just cool.
Now, this has a potential practical application, and that is, as we get towards eradication of poliomyelitis, not poliovirus, poliomyelitis. You cannot, in fact, the authors make a boo-boo, and they say several times that, we're trying to eradicate. The first sentence, global eradication of poliovirus remains elusive. Well, yeah, it's going to be elusive forever. You can't. Only you can eradicate maybe the disease. Forget about the virus. And even Koistja and his TWiV agreed. You cannot eradicate a virus where 99% of infections are asymptomatic. Sorry. I'm sorry. That's something that D.A. Henderson said. Anyway, if you have polio cases in a post-eradication world, you may be able to treat them with this monoclonal because it will neutralize all 3 serotypes.
Yeah.
So it can be therapeutically useful, which is as we know from COVID, is a thing. You can do that. You can treat people with monoclonal antibodies. So
What I would like is, and I don't think that this is possible, is to be able, at least now, is to be able to use this information to create a vaccine that would
Yes.
induce such
would
an
induce
antibody
this.
in people.
Yeah.
Okay?
mm-hmm.
But, someday,
So
maybe,
that could happen.
you
So
got to
the-
create an
You got to
antigen.
create an antigen that does just this, which means
Yeah.
you need to understand how you get
And as I understand
to
it, this is, I haven't looked at it in that much detail, but this is not a linear epitope. This is a conformational epitope, okay?
Yeah.
Right.
it's
So you would
bridging the
have to
canyon, right?
you would have to fabricate something pretty fancy to
Yeah.
serve as an antigen to do this.
Yeah, it's a good point. If you could induce these antibodies, that would be great. Yeah.
Or get 60 oral
60
OPV
inoculations
immunizations
before
every 3
OPV.
months.
Yes.
So
Which
Vincent,
there are,
I have a
there
I
are
have
probably
a pro-
people in the world getting that with the the polio eradication days.
I have a practical question. This goes back to my sort of obsession with techniques. I read in detail the purification,
Mm-hmm.
and I thought of you because I figure, and Alan, I figure, I can see you guys doing this. Is this, so this has making a whole cell lysate and then a PEG precipitate, and then ultimately pellet that through a sucrose cushion and then do a potassium tartrate gradient. Is that
Is
Is
for
that-
virus? Is
what
talking
you
about,
do?
Is it talking about virus? Is
Yeah.
That what, no, we didn't do all that. No,
Okay.
we just did sucrose gradients.
Okay.
Or cesium too. You could do that.
Okay.
I like we would put it on, centrifuge it onto a cushion of sucrose so it doesn't smash into the bottom of the tube, right? Because then you can't get it resuspended. And then you take the sucrose and you do a cesium. And if you have a virus, you could see the band. You don't even need to do any
Good times.
Dripping of
So
fractions.
they're doing all this with wild type 2 polio. Do we have to do that in some kind? And they do make a point afterwards of saying that, they destroyed anything that had wild type 2 polio in it afterwards basically in collaboration with CDC. Okay, so, and is this the paper that?
Must have been in a
no.
BSL-3, no?
Well, this is what, this was my next question, is where do you do this?
Well, now types 2 and 3 are declared eradicated, and so you have to have what's called a polio essential facility in order to work on wild type viruses. And there are some. There's one at Tulane, for example. And
I'm guessing there's one at one of these institutions on the author list.
So the FDA does not have one. I know that, much to the consternation of some people.
What about Hamilton, Montana?
I don't know about Hamilton, but the CDC has one, obviously.
Yeah.
So you could do these. And they do say, after this work was done, we destroyed the virus stocks. However, as you know, I have the sequence and I could make these viruses. But I don't have a laboratory, so I wouldn't do that. No. But it can be made by anyone.
Anybody has the sequence.
You know that's interesting, though? This is a whole contentious area right now because the current guidelines are that in 2025, everybody has to stop working with polio except these PEFs, pet polio essential facilities. But it's actually not reinforceable. The CDC can't tell you what to do. So, and the excuse is that polio is a biocontainment hazard, but it's not because it's in wastewater. So what does it matter if it's in a lab?
Every diaper that's thrown away is a potential polio waste hazard. That
doesn't make any sense. Oh, the other thing here that's important.
Well, it's window dressing. It's we got to make sure that the labs don't have it. So, yeah.
So I think the other thing is they say it's probably unlikely that you will get viruses that escape this monoclonal because then they won't be able to bind the receptor.
Yes.
Right? That's very important because, as you know, SARS-CoV-2 was able to escape monoclonals multiple times, but maybe it wouldn't happen for this. Anyway, that's cool. Yeah, I think it's really neat that I was amazed that such an antibody would exist.
Yeah.
Because the reason is, for many years, people said there are 3 antigenic sites on the particle, and now there's a 4th one. So the moral is, if you just keep looking, you can
never
find
know.
things.
Yep.
You never know. Okay.
And through my extreme ignorance, Vincent, with this, because Vincent was recently in Montreal and gave a talk, and we learned all about poliovirus and enteroviruses and how they can recombinate. And with this shouldn't affect any sort of recombination with other enteroviruses in the gut. Would that potentially affect this site? And would that, like, if there was a new virus that had recombined, do you think that this would still give protection, like this
have
antibody,
to use a
this
different
monoclonal
receptor?
So you're saying, can you get recombination within the capsid coding sequence, right? That would change the binding site for the receptor. Then would have a different receptor as well, probably. But all of the recombinants that we see maintain the poliovirus capsid. There's sometimes the recombination site goes in a little bit at the ends, but you don't see these mosaic recombinations where they're bits and pieces of different viruses, which is what you would need for your idea, right? But just because you don't see it, I mean, we don't look really, so.
Doesn't mean it couldn't happen.
Doesn't mean it couldn't happen, yeah.
Okay.
Never say never. All right, a paper today is in Cell Reports, blocking of Ebolavirus spread through intercellular connections by an MPER-specific antibody depends on BST2 tetherin. Somebody printing?
No, I think
That
it's a
sounds
truck
like
backing up
Oh, it's a
that might
truck.
be-
someone's
That's funny.
truck.
The truck sounds like a printer. I'm thinking the dot matrix
here.
printer. Who has those anymore? Alan probably has one.
No, I actually do not have a dot matrix printer. Although I'm about to get rid of a radio that has vacuum tubes in it.
Oh, don't do that.
Oh,
Well, I don't use it enough.
You can use it as a hand warmer. Turn it
got
around
a
and
few-
use it.
I got it a few years ago, and it's lovely, and I had it overhauled, and it's just a gorgeous radio, and I just hardly ever use it because it's not really the kind of operating I
As
do.
you know,
So-
high-end stereo has tubes, and it's
So I'm finding a new home for it.
good.
The reaction reminds me of, I told someone, this was when I was in Georgia, I was getting my kitchen remodeled, and it had the bright red Formica with the metal sharp, trim around it. And this person was like, "No, that's classic. You have to keep that." And
That's
I
right,
said,
that's
"I will
right.
give you a piece. In fact, you can take it all." But yeah.
Right.
All right, so this, a lot of abbreviations. We'll try and explain them. But this is Rodrigo Santos. I think there's just one first author, right? And
Yes,
then
I think so.
two
Yeah.
corresponding authors, Jim Crow and Alexander Bukreyev. These
Jim
are from
Crow.
University of Texas Medical Branch at Galveston, which Rich and I visited some years ago, Vanderbilt University. And so this is all about Ebola viruses, which many people have heard of.
Yes.
There are 6 known Ebola viruses, and 4 of them can make people very sick, including Ebola virus, Sudan virus, Bundibugyo virus, and Thai forest virus. And the viruses, as you know, are filamentous, long filamentous particles with negative strand RNA genomes. There's a membrane around them. They have a glycoprotein inserted in the membrane. And the glycoprotein, of course, mediates attachment of the particle to cells. And there are multiple attachment factors, which we would normally call a receptor, but here they call them attachment factors because there's more than one. One of them at least, TIM1, is a receptor for phosphatidyl. What is it? What is the phosphatidyl? Shit, I forgot. Let me look it up. Phosphatidylserine.
Phosphatidylserine.
So it is, membranes have phosphatidylserine in them. So that's how Ebola virus attaches. Then it's taken up. So there's not a specific interaction like poliovirus and PVR. And then the virus particle is taken up by macropinocytosis, not endocytosis. It's different. Macropinocytosis is kind of like cellular drinking. Think of a cell needing to drink water, and it's pulling in droplets of water by this process.
Right. Pinocytosis is drinking. Macropinocytosis is big drinking.
Big drinking.
It's
Chugging.
the big gulp process.
This is a Texas thing.
Yes, it's Texas
Macropinocytosis,
style.
Right. So the particle is taken in, and then the macropinosome, which brings the particle in, then goes into the endosomal pathway. Okay, a little bit of convolution here. And in the endosomal pathway, there are 2 proteases that cut the glycoprotein of Ebola virus. They're called cathepsins. In fact, one of those cathepsins can also cleave the spike protein of SARS-CoV-2 should it enter cells by the endocytic pathway. But back to Ebola virus, the glycoprotein is cleaved, and then this exposes parts of the glycoprotein that would not normally be exposed if it weren't cleaved, including the membrane proximal external region, which is MPER, which is in the title. Okay, so MPER is a part of the glycoprotein near the membrane. And then after the glycoprotein's cleaved, it can now bind a receptor in the endosome called Niemann-Pick C1 protein, NPC1. This is a protein involved in cholesterol transport in and out of the endosome. It's very important. And there are kids who have defects in this gene, and they die before they're teenagers because the cholesterol is really important, and they can't properly sort it in the cell. But it also happens to be this intracellular receptor. When the cleaved glycoprotein of Ebola virus binds NPC1, the viral and the endosome membrane fuse, and into the cell goes the viral RNA, the RNA protein complex. So it's very cool because it is not happening at the cell surface, this fusion. It's happening from within an endosome, but it's not low pH triggered. It's triggered by binding to this fusion receptor. So it's a brand new.
And it doesn't get into the endosome in the way we normally think of viruses getting
right.
into the endosome through
Gets in through
endocytosis.
these-
It kind of takes a side door through macropanocytosis.
All right, so that's entry. So now it's all you wanted to ever know about entry of Ebolavirus.
Even more.
Even more.
But what is really interesting in this paper is how the virus particles get out, right? So we think, if you think of an envelope virus, you're thinking, oh, it's budding somewhere within the cell. Let's say it's budding at the plasma membrane. And so the bud forms and then the particle pinches off. And now these viruses can move around from the cell to other cells, right? So they call that diffusion. But there's another way that virus particles can go from cell to cell, and that is actually from cell to cell without going into the extracellular fluid. Cell-to-cell transmission, number of viruses do that. You could, if cells are next to each other in tissues, right? And so if a virus is exiting one, it could fuse directly into another, for example.
And cells
So I
in
have
tissues a lot of them have very, very close associations, including junctions that provide part of a pathway that a virus could potentially take between cells. And that's been observed for a number of viruses, HIV and some others. And the obvious advantage to the virus is if you don't have to go outside, the immune system can't get you as easily.
Exactly. Kathy, you
Exactly.
were gonna
My
say
question. Oh, sorry, that was unless
No,
Kathy
Angela
had a
was.
question.
No, I had nothing.
So I was
Okay.
going to ask, like, for example, with HIV, as Alan mentioned, that it also can go from have this cell to cell infection.
Right.
But can it also have like via endocytosis and bud off, or is this if a virus has one form of like viral release, can it also
Oh.
have cell to cell?
Yes.
I think it can
Infection?
do both.
Can it have both? So
Rich,
it's not
they
specific
include. I'm sorry. Yeah, I think they can have both. Yeah. Rich, they include vaccinia virus here, right?
Yeah, and I looked at the references for that. One of them looked like it was probably a review. Of course, I immediately thought of TWiV 68, my all-time favorite episode, Ode to a Plaque,
Mm.
where we discussed spread of vaccinia, but it's different than this. Okay, I think. Okay, because in that case, first of all, there's two forms of the virus to start with, both of them infectious. There's one membrane particle where that is formed entirely intracellularly. So you have a membrane particle in the cell that's infectious, and it's probably the thing that mediates host-to-host transmission over time because it's a very tough particle. But for cell-to-cell transmission in the cells, that thing can acquire a second envelope and be exocytosed. So now we have the double envelope particle, and that thing winds up being perched. There is a growing filament, or kind of like a pseudopod, out of the cell, a projection out of the cell that is actually actin mediated. Okay, but the particle that is being transferred sits as an extracellular particle perched on the tip of this thing. So I wouldn't call that necessarily intracellular transmission. Okay, and the cool thing is that thing then is deposited on some other cell. And since I'm into it, okay, I have to complete the story, because with TWiV 68, what was shown is that cells that are early in infection make a couple of vaccinia proteins that make it so that if they are probed with one of these particles, they grow another one of these things and pass it on to the next cell, okay, until the particle encounters a cell that's not been infected and infects that. But at any rate, I'm a little confused about this, and maybe you guys can sort me out, because we're talking about intracellular transport here, but are we talking about
Intercellular.
Intercellular.
Right.
Wouldn't
so-
there have to be a gap junction for that to happen, though? Because if not, then it would still have to leave,
Could.
even if it's
Right.
a small space, right?
Yeah.
So it's not
Well, it
It
could.
could-
still
well,
being exocytosed.
Rich, I think the mechanism that with pox that we talked about on 68 and that you're talking about now, it's almost like a handoff,
Yes,
right?
absolutely.
So the virus is actually outside the cell, but it's
Exactly.
attached to the cell, and then it attaches to another cell, and they pass it along until they find one that doesn't have it yet, and they put it in there.
Right. And so I'm wanting to differentiate between that and what we're seeing here.
Yeah.
Are we in this case, when they're talking about intercellular transport, are we talking about a mature virus particle that is inside the cell, or is it because I don't see a in the case of Ebola, I don't see a membrane particle being inside the cell. I'm confused.
Well, if you look at their
I
graphical
was
abstract,
confused about
right?
the topology too, and in their cartoons, it looks like it's a gap junction because they show a tube between two cells and the virus transferring, and I just.
But they don't
Is that
mention
what's going
gap
on?
junction anywhere in
Right.
the paper. Like
Right.
it's not mentioned. It's completely just like assumed, but
Yeah,
not
I
mentioned.
kept saying, you mean a gap junction?
Yeah.
What are you guys talking about?
Yeah.
And
And
see,
I think
that's
is
not
it-
the way I ever think of it, as any kind
Okay.
of gap junction being involved. So when you said that earlier, Alan, I was
okay.
being skeptical in my own mind, but didn't
Okay.
say anything.
So let's to start out with to make a mature Ebola virus particle. Do I not have to bud through the membrane?
Yeah, in fact,
Okay.
Ebola viruses bud from the plasma membrane.
Right.
So this
Okay.
picture in the graphical abstract doesn't make a lot of sense, right?
Right.
Yeah.
So
It's traveling from the cytosol through a tube to another cell, right?
So if I'm talking about, and they're talking about cell to cell intercellular transport,
Yeah.
if it's not using an intracellular particle, it must be something that's like vaccinia in a way.
Which
on
would
the
be like
surface.
on the surface, and then another cell would come and
Exactly,
very
really
close.
close.
It would almost be like a tube for like a split second, almost
Right.
like a communication,
like
but
that.
not really, because it's
That's the way I
already
have
budded.
to imagine it.
So it's
So, I mean-
misleading. Like
Just to add my 2 cents here, because I don't live and breathe virology like Vincent does. I don't think no one else does either. By the
Other
way,
people here,
we're
other
only
people
gonna-
here live and breathe.
I don't.
Excellent.
But I've been very curious all my life as to why rabies virus takes so long if you're bitten on the foot to get to your brain, and it has to travel through the nervous system, correct?
Yeah, I think it
And
takes
that's
time.
So how does that work?
Axonal
Because-
transport is slow. Well, there's a slow form of axonal transport, and I think that's
Yeah, but
part of it.
there's a gap between each nerve, which is
Yeah.
the synapse. So it has to get out of one nerve, go through a space into the next nerve in order to travel north.
Yeah. I mean,
Right.
that's a naive view, right? That it takes long because axonal transport is slow, right? I'm not sure if that's the case.
So in this case, I was misled by the cartoons, which are up in the graphical abstract and repeated later.
Me too.
And I think that may have been the source of my confusion throughout the paper, where I was trying to figure out how they mean for this to be happening exactly. But
I think they don't
what
know.
you're saying with more like a handoff makes a lot more sense, where there's the membrane virus buds out through the virus picks up the cell membrane, budding out, and then goes to the next cell membrane. And I think, as Angela said, there's briefly a tube between the two, but it's not a gap junction, right? Is that?
That's what's making sense to me.
Well, if you look at
Okay.
Figure 7, they don't have a tube in Figure 7. Okay,
Right.
That's their model, where
Yeah.
it's just
But
virus.
the graphical abstract has a tube.
As a
I
tube.
know, I
so
know.
it's like it's very confusing.
It's a
But-
little.
And well,
I think,
their graphical abstract, part of it is showing two cells, and part of it is showing the viral membrane and another cell's plasma membrane.
yeah, on
so
the
there's
bottom.
a zoomed in
that's,
view.
That's Figure 7 on
So-
the bottom.
Right.
Yeah.
With electron microscopy in vitro, you should be able to see that.
Yeah, well, they didn't use that here. They used mostly fluorescence experiments.
Confocal, yeah.
No, I understand that, but you require a finer look in order to make that decision.
The problem with the image, you have to fix the cells, right? And that may be difficult
Well,
to freeze them at certain points. And so
Also,
forth.
with this virus, you need to be doing this in a BSL-4, right?
Yeah.
Right.
Mhm.
Yeah, one of the
True.
things they do here is to use authentic infectious virus. Anyway, so previously people have...
I've been quiet, but I just looked in the famous textbook, Principles of Virology.
Wonderful
All right.
book. I wonder who
Volume
wrote
one.
that.
Chapter
We
477
should have the author on
has-
sometime.
Yeah.
has some
Yes.
EMs of direct cell-to-cell spread of HIV.
Yeah.
Tunneling nanotubes induced in swine testicle cells by pseudo-rabies virus. So there's a lot of material here we could delve into later.
The
So-
one here is that people previously showed that Ebola viruses transmit from cell to cell. And this paper actually is that monoclonal antibodies, neutralizing monoclonal antibodies, actually can inhibit this cell to cell. And that's their mechanism of neutralization. Okay, that's the main point here. And also that there's a protein in the cell called BST2 or tetherin, which is an antiviral protein. And this can enhance the blocking activities of these antibodies. So let's go through briefly these findings. Now, the first one is probably why they drew a tube. All right, so what they look at here is whether actin polymerization is important for cell-to-cell spread of Ebola virus. So in a cell, just below the plasma membrane, there's what's known as actin filaments. There's a cell is a three-dimensional thing. It's not just a flat thing, but so there's a meshwork of actin filaments just below the plasma membrane. And for things to get in and out, that has to be depolymerized and then repolymerized. So in fact, for vaccinia propulsion rich, they're propulsed on tails of actin, right, from cell to cell. So actin polymerizes and pushes the particle on a long projection which has actin in it. So they ask, does this happen for Ebola virus? And they do a cool thing. They have cells where the actin is attached to green fluorescent protein, so you can visualize it. All right, and then they infect with Ebola virus, and they look to see what's happening with the actin, and they see propulsions from the cell. They call them cellular projections with the green color, so that means it has actin in it, and they could stain it for a viral protein. So somehow the actin is making projections from the cell, and it's got virus particles on it, which is very reminiscent of the vaccinia virus story.
Maybe I missed something, but they say they used Ebola, but then they actually only use the BDBV, which is the
Bundibugyo virus.
Bundibugyo virus, which is another filovirus, but they didn't actually use Ebola, although they say they did.
Yeah, it's right. But Ebola virus is the genus, right? So that's okay.
Right.
Yeah.
They used an
similar
Ebola virus.
And exactly.
Not the
okay.
Ebola virus.
Now, they use a drug called cytochalasin D, which inhibits the polymerization of actin. In other words, making long actins that would propel virus particles from one cell to another. And this abolishes cell-to-cell spread, which is very cool, of the virus. And if they use an inhibitor of tubulin polymerization, there's no effect. So tubulin makes long strands from the nucleus to the plasma membrane, whereas actin is wrapped around the cell. So apparently those actin molecules have to polymerize and make extensions to get virus from cell to cell.
Yeah, and actin and tubulin are the two major components of the cytoskeleton. So
Mhm.
they
Right.
use two different functions.
Okay. So then they say, do you need TIM for this? TIM, which is the entry factor for Ebola virus. So again, a phosphatidylserine on membranes attaches to TIM, and it brings Ebola virus into the macropinocytic pathway. So they have cells that lack TIM. They've knocked out the gene with CRISPR, and they infect what they call donor cells, and they infect with Ebola viruses that are tagged with green fluorescent protein, and then they put those cells on top of what they call an acceptor cell, and then they look in the acceptor cells by flow cytometry for viruses getting in, right, from the donor cells. And if you take TIM out, you don't get any intercellular connection transmission. It needs this receptor, which makes sense, I guess, because the TIM is missing in which cells? The acceptor cells, right? Yeah.
Yeah.
So you need TIM in order to get this intercellular transmission. Okay? It's very straightforward. Okay, now they have these monoclonal antibodies. They have a whole bunch of them that are directed against the glycoprotein of Ebolavirus. And they said, does it do any of these block spread? And the answer is yes. In their spread assay, where they have GFP virus in one cell, then they layer the cells on a second layer of cells, and then they do flow cytometry to look at spread. Some of these monoclonals can inhibit the spread very nicely. And one of them, BDBV, which Bundibugyo virus 223, is a very potent inhibitor of virus spread through these intercellular connections.
And they got this monoclonal from somebody whose mom had given him Ebolavirus every 3 months in order. Oh, no, sorry.
No,
wrong
no.
story.
No, that was the other paper, yeah.
Right, yeah.
But this paper, like the other paper, is open access. And there
Yes.
is a pretty cool diagram of this donor cell, acceptor cell experiment.
Yes.
Yeah. And they do talk about these antibodies coming from various individuals who survived Ebola virus infections.
But they're sorting Ebola virus is what, in a BSL-4, Angela?
You can have, we have a cell sorter in our BSL-3, so I'm guessing they have one in there. Like we have it's just in a hood. It has to be in a hood, even if it's in the BSL-4, or in our case, in our BSL-3.
So it's a small one. Every time I think of a cell sorter.
This one's huge. Ours like takes up a whole, like it has a hood integrated in it, right? Like it's in a BSC, so it's pretty big.
By the way, I saw Angela's lab's 2-photon microscope the other day. It's on this platform. You touch it and it goes like water.
Well, if there's any vibrations in the room or anything, it won't screw up your imaging, basically.
Nice.
So it goes. It's like
very
air
cool. It absorbs
cable.
all the vibrations, basically, right?
Mm-hmm.
Okay, so these monoclonals will block this spread. Remember, whatever this intercellular communication is, you need actin polymerization. It doesn't involve the particles diffusing through the medium. You can block it with antibodies. So that's, mechanistically, what's going on there?
Which is, that's when I was
little
thinking
weird.
initially of gap junctions, and then they're using antibodies. And I'm like, wait, no, that wouldn't work because you'd have to get the antibodies into the cell.
Right.
But it's going.
Exactly. So it's not. But
It's
they
this
still
stalk
continue to say
mechanism
that it's
it's just the
connection.
graphical abstract is misleading. If you access this paper, I recommend skipping the graphical abstract because it confused me.
But even the title here, viral spread through intercellular connections. So they're still
Yes.
implying that there's a connection. And I feel like Rich, I keep on looking at Rich and his mind is going. He's thinking through this.
It's more of a handoff than a connection, but it's not the classical route, the front door we think of, a virus floating around in the medium and attaching to a cell.
It's like
more
opening to your neighbor and like running through really fast.
It's like adjoining hotel rooms.
I'll tell you
briefly
what I'm thinking, Angela. I'm wondering about how these assays actually discriminate between diffusion and handoff, if you want to call it that.
They actually do something.
they,
They
though?
put monoclonals in the medium, they said, to get
Yeah.
rid of diffused particles. I don't know how they mentioned that. I haven't really
Right.
mentioned it, but that's one of they do point that to make sure it's not diffusing particles. We do this, and so that's addressed. Yeah. So one of the hallmarks when you add these monoclonals to cells, what they notice is that the cell membrane is all the virions are trapped at the plasma membrane in the presence of these antibodies. Right. That's how the intercellular transmission is blocked. Right. So you treat cells with these monoclonals, infected cells, and the newly produced particles get stuck on the plasma membrane rather than going to the next cell. And they say this is similar to what happens when cells make a protein called BST2. So BST2, or another name for it is tetherin, is a antiviral protein that's induced by interferon, and what it inhibits many enveloped viruses, because what it does, it binds to the plasma membrane at one end, and the other end binds to the particle, and it just holds it at the cell surface so it can't move off.
It
Right.
tethers it to the cell.
Yes.
Yeah, it tethers it,
Yeah.
and that's what they call it, tetherin. And HIV is susceptible to tetherin. And you may say, well, how come HIV is a thing then? Well, because the genome encodes a small protein that antagonizes tetherin. It makes it get degraded. Otherwise, there wouldn't be a HIV.
It's the virus host cell arms race.
Hmm. So they wanted to know if this tetherin made a difference. And they have BST negative cells. And if you take your monoclonal and try and block transmission in a BST negative cell, doesn't work. You need to have BST tetherin there for the antibodies to inhibit intercellular transmission. BST2
So is that like
required.
the tetherin is telling the particles hold still?
I think so. Yeah.
While this antibody does its work, something
Yeah,
like that.
because we know that that's what tetherin does. It traps particles at the cell membrane, right? And so what they say is, well, probably it's enhancing trapping. Maybe the monoclonal is actually enhancing tetherin's trapping, right?
Yeah, something like that.
I mean, this mechanistically, we really don't know what's going on here precisely. I think that's why they have that funny diagram in the graphical abstract, because they really don't know what's going on.
Yeah, part of it is
Hmm.
they get to a certain point and then it's just
Yeah.
here's the observation and we don't exactly know what mechanism is at
So,
work.
but the other thing is, you would predict if tetherin and the monoclonals are working together, they should co-localize, and they do some staining, and they show that yes, the monoclonal and tetherin and the virus all co-localize on the cell surface by staining. So that's consistent with them all working together. Now, the last experiment I think is pretty interesting. So these some of these monoclonals neutralize infectivity, right? They block infection, and this is the mechanism here is preventing cell-to-cell spread. So they say, what if we made the virus resistant to neutralization by the monoclonal? You can make one amino acid changes in the glycoprotein. What would that do to the blocking of intercellular transmission?
Is that a gain of function?
Mm.
Yeah, sure.
Ish.
Ish.
I
Yeah.
mean, you make things resistant to anything,
Yeah.
it's a gain of function, right?
Yeah.
If you make antimicrobial resistant bacteria, it's a gain of function. If you make viruses, and Alan, you may remember in my lab, people made polioviruses resistant to neutralization by
Yeah.
monoclonals years ago, and nobody called it gain of function,
Not a
even though it
problem.
was. Because that name came up in connection with H5N1 in ferrets, and
Yep.
then ever since then, it's been a bad word, right?
Hmm.
But we digress.
It's okay. That's what we do.
Yes.
Anyway, when they have a glycoprotein of Ebolavirus that is resistant to neutralization with this monoclonal antibody, has no effect on intercellular spread. So this single amino acid change is not affecting the ability of this antibody to enhance the activity of TIM1, right? That's the bottom line there. So whatever the neutralization assay is, it's probably not a plaque assay. It's probably some TCID50 or something like that. It's not the same mechanism as this intercellular spread. Although they do have another antibody where the escape amino acid change does prevent transfer of the virus through intercellular connections. So it depends on the antibody.
Yeah.
For some antibodies, the neutralization is overlapping with cell-to-cell spread, and for others, it doesn't overlap at all. Does it make sense? Did I explain it clearly? I think that's
Yeah.
interesting.
Oh, yeah.
Yeah.
That makes sense.
So obviously, the neutralization escape virus, the antibody is still binding it. It must bind because that's how it's bringing it to keeping it on the plasma membrane of the cell.
So
So what did we learn?
I'm trying to summarize it in my head. Help me out here. So
Well, they actually do it in the last paragraph.
okay, let them
First,
do it then.
Ebola cell-to-cell transmission depends on actin polymerization.
Cool.
Okay, so that's cool.
Right.
Okay. Second, the transmission from cell to cell requires TIM1. Okay, straightforward. Third, the transmission can be blocked by antibodies. And in fact, I didn't mention this, but these antibodies target the membrane proximal extracellular region, the part of the spike that's exposed on cleavage and which is near the membrane. It's involved with fusion.
Yeah, the EMPER, which is in the title of the paper
Right.
for
That's
those
right.
that are.
And fourth, antibody-mediated inhibition of this transmission between cells depends on tetherin. And then, okay, those are the four findings. And here's the key. These findings suggest that some protective mechanisms may not be reflected in antibody activities measured by in vitro plaque reduction tests. Oh, so they do plaque assays
Yeah.
to measure
Yeah, they do.
neutralization.
They
Right?
mentioned PFU in some of the experiments.
So there you go. So the plaque assay doesn't reflect this cell-to-cell transmission. Why is that? Because if you make a virus resistant to neutralization by an antibody, which you measure in a plaque assay, it can still block cell-to-cell spread.
Okay.
I highlighted that sentence, Vincent. That's the only one I highlighted in the PDF.
It's the only one.
Yes.
No, it's
So,
a key sentence.
I have a question about in vivo, not in vitro. In vitro is a monolayer of cells, and when you've got tethering, you've got the virus sort of dangling out into the medium attached to the cells, right?
Mm.
What happens when you've got a three-dimensional organ with lots of cells and the virus is in the middle of all of that? How long is the tether to go from one cell to the other, and how do antibodies get to that level? That's interstitial space.
The tether is short, right? But it coats the cell.
It's
in
in
three
it's
dimensions.
interstitial space we're talking about now.
Yeah. And so, when a
antibody
virus
would
virus
that get
buds,
to that level?
well, why not? Sure, it can get between cells. Can it?
I don't think so.
Can't get into them.
We're talking about antibodies, Dixon?
Antibodies even do get
Yeah,
into
they can
cells.
diffuse anyway.
They can
They
get
do.
into cells, right?
They do.
Mm-hmm.
But they can get into the intercellular space, yeah.
Mm-hmm.
Why not?
Okay, so then the other question is, does this monoclonal antibody look as though it could have some therapeutic value?
Ah, that's a good question. They actually don't mention that at all, which
Yeah.
is interesting because most papers do, right?
Mm-hmm.
Oh, they do
I would-
say these antibodies may lead to new avenues of
Yeah, exactly.
development of treatments. Okay, they do say.
Yeah.
Yeah, because you're looking at something other than a plaque assay, right?
Yeah, they didn't do anything in vivo. I guess the next step would be to try something in vivo, right?
It would be
So
interesting
what's
to
the
take that
animal model for
monoclonal,
this?
which is that virus that's resistant to neutralization by that monoclonal, right? And try and see what happens in an infected animal
Mm-hmm.
when you don't neutralize, but you can still block spread. Would that be
Right.
equally therapeutic, right? That would
Well,
be-
and
Yeah.
so has this monoclonal been tested in any animal model, just with normal wild type?
It's a good
Ebola
question.
viruses?
I don't know.
They didn't mention it anywhere.
I mean, I
Seems
would
like they would have mentioned that.
guess that it has been, but that's tough.
Maybe.
You have to use non-human primates, right?
Yeah, you have to use
That's
non-human
true.
primates. You have to do it in a
BSL-4.
BSL-4, so that's a big,
So
that's an
expensive.
expensive and difficult experiment
Which
to
they do
But
in Hamilton, Montana, for sure.
yeah, it might be worth looking at this and then doing the experiment you just suggested, which is when you get escape mutants, what effect does this have? What portion of the infection is being driven by these types of intercellular contacts, and what portion is being driven maybe by soluble virus?
So I think the point for the listeners is that don't just think viruses coming out of cells and diffusion around. They can go right from cell to cell, right? So that is the key here. And antibodies can block that, which is quite interesting.
Okay.
All right.
So I have
All
a
right.
question. This is
Yes.
not about this paper. Well, I got yesterday when I was looking at the paper in my lab, and obviously they use HeLa cells in this paper, which is why I thought about this, which one of you will definitely have the answer to this question. So HeLa cells are human-derived, as we know, but why do we have to manipulate HeLa cells in a BSL-2 and not in a TC, considering it is a immortalized cell line and other human cells we can use in the TC? So
What's
why
the TC?
tissue culture. So BSL-1, sorry, I should,
Oh.
I take for granted. Tissue culture room, like a biosafety lab level 1. So officially, HeLa cells have to be used in BSL-2. So I'm not, but I don't know why, because people use them in BSL-1, but you shouldn't. But I don't know if anybody has the answer to this. If there's something about HeLa cells that, like uninfected, I'm talking just like, it's just a cell line, right? So
they're cancer cells. They could give you cancer.
But so is like KCO2, HU7, every other L929. There's all
they're
of these cell lines.
They're transformed with a virus, but a lot of other cell lines are
Exactly.
too.
This is,
Yeah, I
this
don't know
is
the
a
reason.
distinction that we don't have, that I've never heard of. So basically, other than the fact that some things with E. coli is BSL-1, and actually mouse adenovirus itself is BSL-1,
Mm-hmm.
everything else that we do it really at a BSL-2 level. So distinguishing between different human cell lines between BSL-1 and BSL-2 is a foreign concept to me, and so I'm wondering if it's some Canadian rule.
No,
Interesting.
it's CDC also in the U.S. has
Yeah,
the
because
same
if I
rule.
don't have, if my
Really?
cells
Yeah.
aren't infected, so if I'm propagating cells, I'll do it in the tissue culture room, so BSL-1, and then if I'm going to infect them, then I can take them to the BSL-2, or I'll just start in the BSL-2 and do it there. But I'm allowed to have them. Let's say I just want to look at, let's say actin. If I want to look at actin in some like L929 cells, I can.
Which are mouse cells.
Mouse cells, yes, or HU7, which are human cells,
Right,
which are also used in the paper we spoke about. I can grow those in my BSL-1, so like a tissue culture room, and then stain them for whatever I want to look at. Like I don't have to have them in a BSL-2. So this is why it's confusing to me where HeLa cells, there are some discrepancies with cell lines, even if they're uninfected, and I'm not sure why. Like,
Did we even have any BSL-1 hoods in the lab, Vincent?
No, the whole lab was
You guys-
BSL too.
You were
They were all the same
polio, though.
containment hood, so it wasn't.
Yeah.
Yeah, we have a
Yeah,
lot
I-
of like tissue culture because we just look at
Okay.
like cells, like T cells, things like
So
that.
what's
but-
weird is that the documentation I'm looking at here say it's a risk because the cells contain HPV-18, but they don't. They only contain E6-7, right? They don't contain the whole viral
probably
genome. So I don't know
Yeah,
why.
I'm looking at another thing that says ATCG general guidelines recommend BSL-2 containment for the broad category covering HeLa, and specify that BSL-2 is for cell lines that harbor mycoplasma or any other BSL-2 agent, cell lines exposed to or transformed by a primate oncogenic virus, primate cell lines that contain viruses, cell lines carrying a part of certain viral genomes, even if whole virus is not released from the cell.
It must be
Okay,
that. Then,
two
then,
words
yeah.
that stood out to me in that were guidelines and recommend.
Because mycoplasma, everything could potentially
and
be
also
infected.
mycoplasma, because
Everywhere.
I grew a lot of mycoplasma containing cells.
Yeah.
Yeah, we're not
completely
supposed to.
unintentionally.
We don't want mycoplasma in our cells.
No, we
No, but they
absolutely
could
do
be.
not want it, but it
Yeah.
just
Yeah,
shows
of
up.
course.
Or Sendai virus either.
Right? Okay.
Mm. Okay.
Does that answer your question?
No,
I think-
probably
No.
not. But
I think it's maybe it could be rethought. The guidelines could be,
yeah,
More of a guideline.
it's more
fixed,
guidelines.
updated. But yeah, okay, not really. I feel
I
like
got
you
a
could
hunch
still
if they
use them.
catch you with your HeLas in BSL-1, they're not going to throw you in jail.
Let's
They'll
read a
throw you on
couple.
a BSL-2.
Let's read a couple.
They'll put a hood on you.
They'll put a lid on you. That's right.
Let's read a couple of emails. Okay. Yeah, Kathy, can you take the first one?
Sure. Adriana writes, "Hello, TWiV team. I'm finally writing to say a heartfelt thanks for all that you do to educate and inform people about viruses specifically, but science generally. I started listening to TWiV at the beginning of the pandemic. I was a school nurse and was advised by a pediatrician that your podcast would be a real help. It was and is. Even though I have been retired for a year, I still greatly enjoy listening to TWiV, and that includes the banter. I am sometimes just hanging in there by a thread in understanding exactly what it is you are talking about. The thread almost snaps, but then one of you says, 'So basically,' or 'in other words.' Then I get the point of the discussion and the primary takeaway. I have learned and continue to learn so much from TWiV. Your podcast made a huge difference during the pandemic when I was navigating the real concerns of my elementary school students and worried parents, teachers, and administrators. I was able to say I do not know when that was really the only truthful answer. I could do that with confidence because you are also saying that, but with additional information on what could be known and what would need to happen in order to know. I've been a nurse in various pediatric settings, including critical care, a refugee camp, and public health clinics over my 50 years of practice. Being a volunteer vaccinator during the pandemic seemed like going out with a bang rather than a whimper, so that is about the only good thing about the pandemic other than now indulging my lifelong interest in infectious disease by listening to your podcast and reading the books and articles you recommend. I've read Immune, A Shot to Save the World, Racism Not Race, Breathless, and I have just started Fever, all the books that were discussed on TWiV. I love your picks and always look forward to sharing your broad interests and passions. Cooking with an Italian grandmother has become a favorite, not only for me, but my entire family. That was a pick of Vincent's. I so appreciate your standing up for science and against misinformation and the flat-out lying for political gain. My husband, a now retired pediatrician, and I are sad and disheartened to see the current trend into anti-vaccination and vaccine misinformation. We count TWiV and Paul Offit among those champions for children whose voices are so desperately needed. Do not stop what you are doing. Speak loudly and boldly. You are needed. Count me as a grateful and committed listener, Adriana. And then I guess this is a quote she's added. Children need to be healthy to be educated and educated to be healthy. Quote, if access to health care is considered a human right, who is considered human enough to have that right? And that's by Dr. Paul Farmer.
I think it's access to healthcare is a human right. Education and healthcare.
Agreed.
Here.
This is great. Thank you. Wonderful.
I really like this bit. I was able to say I do not know when that was the only truthful answer, and that I could do so with confidence because of our example.
Yeah.
I mean, that's really important.
Yeah.
Yeah.
Mm-hmm.
Dixon, can you take the next one?
Yes, I can. Rachel writes, hi, TWiV. Hope you're enjoying the cooler weather. I'm an intern at a hospital in Queens and recently diagnosed my hospital's 4th case of West Nile virus. All cases were from this September. 3 out of 4 of the cases had central nervous system involvement, and one of the patients died. I've only been at this hospital for a few months, but from the reactions I'm getting when I tell attendings involved in the case what the diagnosis is, it appears most of them have never even seen West Nile before. I wanted to write because you have so many clinicians in your audience, and I'm sure many of them also don't think of West Nile immediately. But I think it's an important diagnosis to keep in your differential. For patients with fevers and flu-like symptoms and or encephalopathy, where they can't find another obvious cause. I don't know if this year is particularly bad, but with the global temperatures rising, I'm sure we'll be seeing more mosquito-borne viruses up here in the Northeast. Rachel. Indeed. That's how the first outbreak in New York started, too.
Yep.
Let's see. This is about consciousness, so that's for Rich.
Wake up, Rich.
Right? There's a link to a YouTube video on how did consciousness evolve. I have not looked at this, but oops. Hang on.
And this is from
Sorry.
Jeannie.
I have not looked at this yet, but I will. It's a talk by a guy named Nicholas Humphrey. It's an RI lecture from Nicholas Humphrey, who has also written a book on this topic. Hope you have a wonderful day. Here in Inuvik, Northwest Territories, Canada, it's 2 degrees C and dark and getting darker. We will lose the sun in about 2 months and lose about 15 minutes of sunlight. That's from Janine Mills, the knitting crone, she calls herself.
Wow.
I need to look at this video. Thank you very much. I will.
Knitting sounds like a good thing to do in Inuvik.
Yeah.
It does. Is that the farthest north we've ever had?
I'm gonna.
True.
look
Have
at
you ever
this.
had letters from Nunavut? Because that would be more north, like
I have
Iqaluit.
not.
Don't
Or
recall.
Tool.
Okay.
We've
Never
gotten them
Thule
from
either.
like northern parts of Scandinavia, haven't we?
Svalbard.
Maybe.
Svalbard. Yes,
I don't know.
yes.
I don't
That's
know
right,
if we've
that's
gotten a
right.
letter from there.
We've talked
A polar
about
bear, maybe?
Now, we had a pic of this seed repository in Svalbard,
Yes,
right?
yes.
Is that where it
Right.
is, Dixon?
Yes, it is. Yes, it is.
Okay, let's see. That was Janine. So, Angela, can you take the next one?
Yeah. Michael writes, Vincent, I'm an assistant research professor at Penn State, and as part of my extension appointment, frequently teach the public about black-legged ticks and Lyme's disease, since they are so common in Pennsylvania. I also co-host Arthropod, an entomology-themed podcast that covers all aspects of the discipline. I knew West Nile virus is present in Pennsylvania, but didn't know much about it since I'm usually so focused on ticks and Lymes. So I was excited to listen to episode 1031 of TWiV, Death on the West Nile. Your discussion of the Gervais et al. 2023 paper was so interesting, and I thought we should cover it on Arthropod. So in the most recent episode of the show, we did a deep dive into West Nile virus, where I laid out the history of West Nile virus from its discovery in Uganda to its spread around the world, and how different mosquito species with different host preferences amplify West Nile in birds and then transmit it to humans. Signs and symptoms of West Nile virus infection in humans and horses, and capped the show off with a discussion of Gervais et al., which included background on things like what is an interferon, since we don't usually cover such topics. Thank you for discussing the paper on TWiV and providing the inspiration for me to learn more about West Nile. Best regards, Michael Skvarla.
I swear we had
Michael
him on.
is a PhD at Department of Entomology
Penn State.
at Penn State.
I
Yeah.
thought we had him somewhere on one of our pods. Because the arthropod, it's a great name, right?
It is
Arthropod.
great.
I'm
Yeah.
gonna go listen to this. I need to. I
Yeah,
need
they're
to
up
go
to
find
episode
this.
146, so they're sustaining
Wow.
it.
So,
Yeah, I'm betting that, Alan, you're really jealous of that name.
I am very jealous of that name and that topic. That's a great thing, and also the fact that their October episode was a deep dive on the 1986 movie The Fly, which is perfect for Halloween on an insect.
So,
podcast.
Vincent, didn't
Great.
we do our first TWiV on West Nile?
Sure. TWiV 1, yeah. West
Yeah,
Nile
so
story. Yep.
Number 1. And
Mhm.
I wrote a book with your help
Yep.
called The West Nile Story. So we
Yeah.
recommend that to them as well.
Let's see.
Number 1 and then 1031.
Right.
I have it right
Took us-
here. West Nile
There
story.
you go.
I think
There
you
you
can.
go.
Can you buy this on Amazon?
You can, and you can
I
buy a
think so.
Kindle version as well.
Look at
Dixon,
this.
did you
It's-
send
it's-
us copies of your book yet? Your new book?
I have not done that. Remember, good friends buy books of
No.
authors.
That's
at the
true.
show.
Run the yes.
Let
Dixon,
us
you
know when it's
autographed
available.
this for me. Look.
I did. Well, you were a big help. You were a very
No,
big
I
help.
said, without your help, all the virology would be suspect.
Correct.
West Nile
Probably
story.
still is.
This is a,
No,
this
it was
good
a
book.
lot of fun.
Remember.
I had a lot of help with that book. A lot of help.
This is the way it begins. Chapter 1, The Mysterious Stranger.
Yeah.
I would love to read books on the radio. St. Louis Blues. 6 people were admitted to Flushing Hospital in Queens, New York, between August 12th and August 23, 1999, with similar enough symptoms of high fever, altered mental status, and headache to make it probable that they were suffering from the same thing. The fact that they all came from northern Queens made it even more probable. But suffering from what? Oh, it's drawing you in, Dixon.
Yeah. Great opening.
It's very good.
So I Googled for his name on Microbe TV and didn't find anything, but I did find a link to the Purdue newsletter from entomology that includes something about Mike Skvarla, who is a graduate of Purdue. And the episode also, the newsletter has a discussion of the 30-second bug bowl, which is a Purdue thing. And anyway, recognizes
Sounds
his
delicious.
podcast and so forth. Yes. But hasn't been on Microbe TV that I could find.
Here
We should
we go.
remedy that.
Here's the episode.
Oh, you found it?
Jonathan from the podcast Arthropod joins the Twipperati. Jonathan, what's this guy's name?
Oh, this is Mike.
Michael.
So yes.
I guess.
Is it Jonathan Larson?
Let's see. Let me go.
Because that's also
yeah, Jonathan Larson. Yeah.
Okay, because he's also a Purdue grad.
Oh, he's in the
Okay.
same podcast. It's just two different
Yes.
people that
Right.
do the
And they're
same
both
one.
Purdue
Yep.
grads. They're both mentioned in this newsletter.
There you
Okay.
go.
Cool.
All right, my memory served me well. That episode was called
Yes.
Entomophagy.
Cool.
Oh, very nice. All right, what are we up to here? Rose? Who has
Is
not?
a rose, is a rose.
Alan, have you read?
I have not read one yet.
Please read.
Rose writes, I just finished listening to TWiV 1047 about avian flu and had a small anecdote to relate about rhinorrhea in chickens. In 2005, I did a story about avian flu and the biosecurity measures taken by commercial growers. For that story, I interviewed veterinarian Donna Carver from the poultry department at North Carolina State University, who explained that when chickens get runny noses, they don't have Kleenex at their disposal. If you walk into a flock and you see dirty feathers over the shoulders or the wing area, then you probably have a respiratory disease because they're wiping their eyes and noses on that and the dirt from the house sticks to it. She told me. Now, in our household, if my husband and I are feeling ill, we'll announce it to the other by saying we have dirty shoulder feathers. Fascinating show, as always. Rose is an RN, MPH, and a health reporter. Very, very cool.
That's an
That's
episode
so
title.
Wait
they
a
wipe
minute.
their
somewhere.
noses with their wings
Right.
like on their sleeves.
Yes.
Wow, that's just funny. I
Good
think
stuff.
this beginning, a small anecdote to relate about rhinorrhea in chickens.
Rhinorrhea in chickens. We get the
Uh,
best letters.
I will
Yes.
come back to this, but if I'm not mistaken, I had lunch with Rose
It might.
Ah.
You might have.
Oh,
Yeah.
wow.
when she visited Austin.
Yep.
I gotta.
Check it out. All right.
check it
So, I think I'm the last one, right? Everybody's read.
I think so. Yes.
Simon writes, whilst I don't keep up with as many of your shows as I did during the pandemic, I do still make a point to listen to as much as I can. You have a great community, and the work you all do for science communication is so important. Anyway, for the last year, I've been participating in a study by Seramune, mostly in SARS-CoV-2 antibodies. It's interesting in a few ways. First, you do your own blood draw and send it in by USPS. A neat little device you stick on your arm, click a button. This causes a small puncture. Blood drips into the attached vial, not VILE, but
VIAL. Yes.
Then you peel off and put on a Band-Aid. I don't know what the peel off refers to.
You peel off a Band-Aid from the strip that it comes on and you put
see.
it on. I think that's what that means.
Second, they do this every 6 months and profile antibodies to about 50 or more epitopes across what is S and N? Across S and N.
First, I
yeah.
thought south and north, but that doesn't make sense.
Maybe,
No.
well, maybe northern and southern hemispheres.
South and North. Anyway, they share your
maybe.
own results plotted over time and also post wider results. Third, they also test for half a dozen or so mother antibodies. What is a mother antibody?
Maternal?
How would they know it's maternal?
Oh,
though?
yeah.
That-
That does. I don't know.
Yeah, but it must be mother.
Other antibodies, maybe?
I don't know. Maybe.
Some
Maybe it's a
other-
typo and it's other antibodies.
Maybe it's, yeah, or some half a dozen
Yeah,
or
okay.
so other antibodies. Yeah.
Other
Mother-
antibodies.
mother
yeah, we like
antibodies.
that. Yes.
Other.
Yes, Rose and I had lunch together about a year ago. Hi, Rose.
Oh, S and N, the SARS-CoV-2 proteins spike.
Spike and N.
Ah,
okay.
okay.
Hey, Angela, can you hear me? Cool. So let's just go on. All right. It's a ending for Simon. Anyway, you should check them out. They also apparently just had an article published in Long COVID Biomarkers. Keep up the good work. Simon, hot fall in Bay Area. All right. Time for some picks of the week. Dixon, what do you have for us?
Well, I'm going to concentrate most of my picks from now on issues related to the environment and how it's not doing as well as it should be. And the one and of course, we're all interested in science education. I don't know if anybody else is aware of this or not. climatescience.org is a wonderful entry-level website for people who are confused about what's going on out there with regards to the naysayers and the sayers and everything else. When you look at the simplicity of their explanations, they're not dumbing the science down, but they've got great graphics that almost appeal more to kids between the age of 5 and 10 years old than they do for older adults, which I think is a good thing because then everybody gets it. Because you have to sit with your kids and see what they're watching anyway, so you have to monitor their cartooning. It's a great website. I watched several episodes, and you have to take courses. Some of them are very short, others are a little bit longer. And I think it has. I'm going to go through the whole thing and look at it because I think it's just something that's been missing in the public's image of what science is. And science is, at some level, very understandable. And this website makes it very, very understandable.
And this is a great approach with like kids' cartoons
That's right.
because the parents who are too embarrassed to ask questions because we've
Yes.
gotten old and obsolete about
Mhm.
that
Yeah, you're
are going to say, oh, okay, well, I'll watch this because my kid is watching it. I want to see what it's
Right.
about. And
That's
then
right.
they will
That's right.
actually
That's
get
right. That's
the
right. That's right.
concepts. Yeah, that's great.
Nice. Kathy, what do you have for us?
I picked a website that I thought of in August that I would pick it just before Halloween, and I didn't end up being on the show just before Halloween. So it's a website that a friend of mine put together for someone that he knew, and that someone that he knew is Richard Harris, who passed away, unfortunately, just last spring. But he has this amazing art collection, over 3,000 objects, the theme of which is death. And so the webpage is very well put together. You can look at all kinds of different categories. What I like is if you mouse over any of the images, it tells you the size and the media and who the artist is.
It's
And
amazing.
the collections include Asian, contemporary, Dance of Death, Day of the Dead, ephemera, ethnographic, pop culture, which, for instance, has a Grateful Dead image on it. Cultures of Death, a Mexican experience, traditional. So, and this person had his art shown in several different shows. That's described in the About section. For instance, the Chicago Cultural Center, another one, where was it? Somewhere international. Oh, in London. Yeah, the Welcome Collection in London. So it would have been a little more appropriate just before Halloween, but I actually know somebody who's going to a Halloween party this weekend, so maybe it's not too late.
You're also pretty close to the Day of the Dead, which is good.
Yes.
Yeah, that's right. That was
Oh,
not
and
long ago.
Richard Harris passed away in March of 2023. He must have been so pleased.
Right.
Now he's part of his own exhibit.
I'm sorry that
Right.
he's gone. This is a very cool collection.
Right.
I really like this, Kathy. This is great.
Yeah.
Wow.
Yeah.
So my wife found a title of a book that he might would have, if he were still alive, I'm sure he would have added it to them. It's a wonderful title. It says, "I'm dead. Now what?" You know, that's.
Rich, what do you have
Dear.
for us?
New sci-fi pick. Some books I'm reading. It's a series called The Murderbot Diaries by Martha Wells. They're fun because these are like novellas. So it's a few hours each that are a read. And there are 9 of them published currently, with 1 anticipated. The link I have here is to the Wikipedia description. And I can just read from that. The series is about a part robot, part human construct designed as a security unit, that is SEC unit. The SEC unit manages to override its governor module, thus enabling it to develop independence, which it primarily uses to watch soap operas. As it spends more and more time with a series of caring people, it starts to develop friendships and emotional connections, which it finds inconvenient. So it's really very entertaining. I really enjoy them. I guess in some ways, it's pings on my consciousness theme, right?
Yes.
Because this is something that, you know, an entity that was created, you know, partly to serve humans, had to have governors put in place because of its biological components to keep it from being too human. And it's now overriding that stuff. Very interesting.
Interesting. That is interesting.
Cool.
Yeah. Alan, what do you have for us?
I have a podcast. I actually don't listen to a lot of podcasts because I don't have a driving commute or anything, but I came across this one and got totally hooked on it after the first couple of episodes. It's an audio drama. And it is the work of, there's one guy who does almost all the voice acting in it, and he and his partner do the writing. They have occasional bit parts by other voice actors. I had intended to pick this right before Halloween, but we're close enough. This is Old Gods of Appalachia. And the central concept behind it is, what if, by digging into the black centers of some of the oldest mountains in the world, humans have unwittingly released something that should have been left buried. So it combines. Well, no, this is far more Lovecraftian.
More like a Balrog, huh?
Yeah, it combines gothic horror in a Lovecraftian theme with a fictional version of Appalachia that includes bits of real history and real Appalachian folklore. And as having been raised in a Southern family, I found this really resonated with me, but I think it's kind of universal. It is a horror anthology podcast, and as such, may not be appropriate for all audiences. Listener discretion is advised, as they say at the beginning of every episode. But I will put an asterisk on the horror aspect. I'm very picky about horror. I don't like psychological horror or jump scares. But cool monster horror is awesome. And I would describe this more as spooky than scary. So give it a try. It's a hoot.
Cool.
Excellent.
Horror. My pick is a website called Poetry Foundation. I was just looking for some poems, which short poems I like, especially when they're just perfect, right? And so I found this website where you can have it email you a poem a day if you'd like. But it's a really much more than that. It is actually has the whole archive of Poetry Magazine. And so this website evolved from the Modern Poetry Association, which was behind Poetry Magazine, and now they got a endowment to keep this going forever. And so there's all sorts of poems and articles about them, articles about the poets and so forth. Poem of the day. Let's see, today, Fourth Wall Arpeggio by A. Van Jordan.
Yeah.
Lately, my friends ask me out of love, have I written about my mother who suffers under the storm of Alzheimer's disease? And I tell them, I don't write about my family, never directly at least. To write this poem seems so out of character. And it keeps going. Anyway,
Who's your favorite poet?
Oh, it's probably Robert Frost.
Yeah, that's what came to my mind.
And then E. Cummings behind him.
Right.
My current one is Billy Collins.
Really?
Yeah. Any poem by him.
Billy Collins. Let's see if there's any here.
Oh,
Billy.
yeah, on your site, but yeah.
My site. Yeah, there is
lots
Billy
of
Collins
books. Here. He reads his own poetry. He does fantastic poetry readings. He's just amazing. Really funny.
Most popular poet in America.
Robert Frost?
No, Billy Collins.
Billy Collins.
Oh, Billy Collins.
Okay.
Robert Frost is dead, you know.
But of course.
Oh, look at
But
this.
he's still popular.
Look at this. He collaborated with Amy Mann.
Mm-hmm.
So you may say, who is Amy Mann? Well, she's a well-known singer who got her start in Boston when I was there from 1979 to 81. She was in a band called the Young Snakes, a punk band. And I went to see her one night. There used to be, I don't know if it's still there, an Italian diner called Cantone's, okay? And it had booths with vinyl on them, you know, and Formica tabletops. During the day, it was an Italian diner. And at night, they had punk rock. And you sat at the diner things, right? And the band was at the other. So one night we went, me and Ihor Lemishka actually, went to see Young Snakes. And there's Amy Mann, who years later becomes big as an independent. I was trying to tell someone when I was in Boston a few weeks ago. I couldn't remember her name. There it is. Billy Collins and Amy Mann. Cool.
Wow.
All right, that'll do it for TWiV 1059. You can find the show notes at microbe.tv/TWiV. You can send us questions, comments, picks of the week to twiv@microbe.tv. If you like what we do here, please support us. We are a 501c3 nonprofit. Your contributions are U.S. federal tax deductible. We need your help to do this work, getting the right science out there. Dixon de Pommiers at triclinella.org, thelivingriver.org. Thank you, Dixon.
Thank you. As Kathy would say, I learned a lot.
Tell me one thing you learned, Dixon.
I learned that they didn't really find out how Ebolavirus gets from cell to cell.
That's good. That's good. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at turbidplaque.com, alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
And Angela Mingarelli is at McGill University, Immune Vet on X. Thank you, Angela. And by the way, I was just in Montreal this past week with Angela for a couple of days, and we recorded a TWiV there. It was really cool. It should be released one of these weeks. Look for it. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week with another TWiV is Viral.